Yeast haploid cells can exist in one of two mating types, either A or alpha. The two mating types can mate with each other to form a diploid cell that can undergo meiosis. And you can see that cycle up there. So it starts with the asopores that germinates and buds and goes through a haploid phase. Or on the other hand, you have the A, which germinates, buds, goes through another haploid phase. Both the alpha and the A will fuse to make a diploid cell through budding, which results in 4 meiotic products or haploid spores. In this particular experiment, we look at the adenine synthesis and the mutations that can arise from that. So ADE stands for adenine, and any mutations of this gene encoding the enzyme will inhibit the synthesis of adenine, thus resulting in the conversion of cream-colored cells to red cells. So we look at this pathway, and if there's a mutation to ADE1, which is highlighted in red, or ADE2, the mutation will lead to an accumulation of the red pigment, as we can see here. So the first 2 plates were the original testing. We tested unknown 2 and tested the mutant and wild type strains. And then we kind of got a little cross-contamination in the HB2, HBO kind of regions. It's a little unclear as to if it was fully red or was rescued by the white cells. So I went ahead and re-isolated those 3 strains and was able to distinguish HB2 as having the accumulation of red. And then going back to the plates, the proper way to culture this in this experiment would be this 3rd plate here, which is the one that I did. So you take your unknown strain and go one way, and then you cross that with your known strain, so the HB2, HBO, HBA, and then through that you'll be able to see the proper results. Okay, so as we just talked about, we will be culturing the plate. So you're going to take your unknown strain with a clean strain. You're going to divide it into 4 sections, then correctly label so that you do not get the types messed up. You want to indicate the direction that each type will be swapped so that when you come back to look at it later, you know exactly what you're looking at. And this is pretty much what the completed plate labeled should look like. So you'll go ahead and flip this over, then take each labeled strain, the mutant and wild strains, then you will swab those with a sterilized loop using the correct techniques as described earlier in this semester. You want to swipe in the direction of the labeled plate and then grab a new sterilized loop and swab your unknown and follow the same procedure, but make sure that you're following the correct direction.
Dude, my grade is so bad for this class for general genetics.
Really? Once you seal your plate, you want to properly dispose of your loops and then finally label it with your name, the class, and the date.
We need 2 grams to 200 milliliter beer.
And when that is all complete, you will take a walk down to the incubator and store it overnight at 25 degrees Celsius and check your results.